Okay, we're going. I don't know about the audio yet, but I'll configure it on the fly as best I can. So.
Create a new science and pharmacy lecture series, and that is the origin of doing things this year. So with that, I also want to recognize and thank Phil Nudelman and the late Lieutenant Nudelman, whose generosity and vision made this lecture series possible. The Nudelman family is here. Mark and Carol and Phil, to one minds great to. Yeah, we are so grateful to your enduring support of the school. Appreciate your generosity, and thank you so much for your continued support. So at this point, I want to welcome Professor of Pharmaceutics, who will introduce our tonight's guest.
Wonderful to see such a big turnout. Good afternoon, everyone. I'm Josh Giacomini, professor in the Department of Pharmaceutics, and I'm going to be co-moderating the session with Professor Joanne Wang when we come to the question and answer session as well. And it truly is my pleasure to introduce tonight's special guest and a longtime friend, and our inaugural speaker of this seminar series, Dr. Catherine Giacomini, who is the dean
So at this
of the
point, I
School of
want to
Pharmacy
welcome
at the
Professor
University of California, San Francisco.
Professor of Pharmaceutics, who will introduce
Kathy and I
us
go
to
back a long
the
ways, from UCSF when I was a postdoc there. And over the years, I've seen her work and her research. And she has become a distinguished scientist and academic leader who is widely recognized for her research on membrane transporters, and their genetic variation. Good afternoon, everyone. Drug disposition and response of drugs as well in the department of causation of disease. And she's also going to be co-moderating characterization of key human transporters. When we come to the question, Giacomini has helped advance the field through broad leadership, including co-founding the International Transport Consortium, which my pleasure is introducing our special guests that have emanated from that consortium in high-profile journal like Nature Drug Discovery and so forth. And she also has led the Pharmacogenomics Global Research Network. And not only that, on top of that, Dr. Giacomini's contributions to science and education have been recognized through numerous honors at the Academy of Medicine, which is a very prestigious honor as well. We're grateful to have her with us this evening as we launch this new lecture. Her work reflects the kind of rigorous, thoughtful science that connects discovery to better care, and her leadership has helped shape the future of pharmacy in both research and education. The Science in Pharmacy Nudelman Lecture Series came to right there. Dr. Anirban Basu, I don't know whether he's here today. And of course, Daniel Webb. Where's Daniel? He's here somewhere. There he is at the back. You know. The committee would also like to acknowledge the excellent logistical help provided by the advancement and the IT team. Eva Eric, who's right at the back there, who organized this whole thing. She did the heavy lifting. And Jason Williams, our IT person, okay? And their tremendous help. And I'd like to say that, we've had a huge interest in this event because of Dr. Kathy Giacomini's work. I think we have 80 people sort of in person and more than 150 people online, So now please welcoming Dr. Kathy Giacomini.
This is going to be challenging for me because first I got to turn it the right way. I talk in here and I advance this way. Yes.
That's right.
And I can point.
You can point as well.
Okay, I got it.
And the back and forth.
Okay. At the end of this. Okay, so first of all, big thank you, Josh. And I also want to thank the committee that you just acknowledged. And I want to thank the wonderful staff who have taken care of me all day, and Joanne Wang, and I want to thank Jay as well, and all of you who I've sat and talked and enjoyed the day with. It has been a spectacular day, learning about all the different research that's going on here at University of Washington, which is really a premier pharmacy school and a premier research organization in the country. So it's been really fun learning about that. All right, so I'm going to talk about transporters, which, as Josh said, has been how I've devoted my career, and how they matter for health and disease. And I'm going to start my lecture, because there are students here, with early studies and what we did way back then, before there was molecular biology. I'll move to my studies focused on deorphaning, or learning what genes in the genome, that 20% of the genes in the genome, we don't know what they do. So one of my jobs, I feel, as a transporter biologist, is figure out what those orphan transporters do that no one knows. So I'll be talking about that. And then I'm going to talk about the blood-brain barrier at the very end, time permitting. So we'll see how that goes. Okay, so what's a transporter? So molecules, drug molecules, for example, have to cross membranes. They have to get to your heart, they have to get to your brain, they have to get to wherever they're supposed to be working at. And to do that, they often need transport proteins. And the These are cartoon representations of transporter proteins. They're really more complicated than that, but I'm just highlighting that some transporters may involve exchange, like the transporter goes this way and another molecule goes that way. Others may just be a simple channel almost, where the transporter just moves across, the molecule just moves through this protein. And others can involve two molecules moving together, the drug and maybe sodium. So there are just different mechanisms by which they work. And I'm not going to talk about all those different mechanisms, but just know that they can work through various mechanisms. So when I started my career, we were in a pre-molecular biology era. There was no cloning, there was no recombinant DNA, there was just sort of characterization. And then we moved to the molecular biology era. From there, the human genome, we didn't have a human genome when we had one, but we didn't know what it was. It got sequenced, and everybody can remember around 2001, the human genome was published. And before that, those years preceding it, we were learning about the human genome. And then finally, I think we are now in an era of technology, computation. We all know we're being influenced by AI every day. So technology, computation, and engineering biology. Not just observing it, but engineering biology, and talking to the people here at University of Washington in the School of Pharmacy is very clear that we're into that era here as well. So I'm going to tell you a little bit about this. To the students here, technology is very important. If you need to often be using the latest technology and the latest innovation. So early adopters do well in science. You can't just learn something when you go to graduate school and then keep using that the rest of your life. Science is changing at a rapid pace. I don't love technology. My husband, he is always getting a new car, and showing me how it works, and I just noticed that, oh, it's red. So I don't have that love for technology, but I collaborate. That's the other thing about science, it's increasingly collaborators. So if I need a technology, I understand what it will do for me, and then I collaborate. So that's an important message I want to give today. So back then, way back in the 80s, I was working in the area of pharmacokinetics, and pharmacokinetics had just been developed. And I was working on a drug molecule called disopyramide. It now has a black box warning on it. But its concentrations were declining. These are the plasma levels. They were declining with time. It had a half-life. We were learning all of those concepts. But it was also being eliminated in the kidney. And that interested me. Like, how was it being eliminated in the kidney? What was happening in the kidney? And we realized that they were probably carrier molecules that pushed that drug out into the kidney, but we didn't know what they were. So I began to ask those kinds of questions. How is disopyramide actively eliminated by the kidney? What's going on there? What molecules are moving disopyramide into our urine? How are we getting rid of it? Look at the levels going down. And so at that time, the newest technology is, again, we're pre-molecular biology. So the newest technology was taking the kidney and dividing it, homogenizing it, and dividing it into the apical membrane and the basolateral membrane. These are the two membranes that the drug has to get into that renal tubule and cross two membranes, one membrane and then another. And we could separate those membranes. And we could learn about how disopyramide was transported across this membrane and that membrane. So I went down the hall and learned how to make these membrane vesicles from kidneys, because I was in a pharmacokinetics lab, and that guy couldn't do it, but someone down the hall was able to do it. And he was studying glucose transport, how sugar was getting out of the kidney and back into the kidney. And I thought, well, I can apply these methods to how drugs are getting out in the kidney. So we did that, and we published a number of papers. I'm just showing you a few of them on using those membrane vesicles, the pH-dependent transport of organic cations in the brush border membrane, et cetera, et cetera. So we were really learning about how these transporters, so-called carriers, transporters, worked. And we learned a couple things. One, that if you were a basic drug, you were transported by the same transporter that another basic drug was transported by. And that one basic drug would inhibit the transport of another basic drug. But if you were an acidic drug, you used a different transport mechanism. So we began to characterize, and we learned those characteristics. Saturable, very specific, charge-specific. Bases didn't interact with acids, competitively inhibited. So we learned all that. We learned the driving forces for these transports. But we didn't know what they were. What are they? You've got this gamish of kidney vesicles. What is it that we were studying? And then we came upon, of course, a big question. We needed the molecular biology era to say, what is the molecular identity of the kidney and liver transporters for basic drugs? That was a question I asked back then. So I had to go do a sabbatical. It was great. I went to Switzerland. I learned molecular biology because that's another thing in science. You have to retool. There are things you soon get dated. So you either go on a sabbatical, you go work in somebody's lab, you have to learn the new technologies as they come. And so I did, and we cloned the first human organic cation transporter, really the first human transporter, 22 family, which I'll talk about. And as you can see, these are the amino acids, so we learned what it was, and that was, I would say, one of the most exciting days. I've been looking at this gamish in the kidney. I've been watching it take up drugs. I've been watching drugs inhibit each other. And now I knew the molecular identity of that transporter. And we were celebrating then. And Juan, right here, was in my lab. And we were also interested in nucleosides, purine nucleosides. And Juan cloned the first human purine nucleoside transporter. We learned its molecular identity, and we were also celebrating. So we learned what these transporters look like, and what their molecules were like. And from that, we learned a lot more, and I'll get us into that. So one of the observations we made, and this is another message I want to give people, look at your data. Spend as much time looking at your data as you do generating your data. This is what I tell my students all the time. So Lei Zhang in my laboratory said, you know what, we cloned this organic cation transporter, and our competitors published a month later the organic cation by 1 month. And Lei pointed out to me, you know what, there are 554 amino acids that make up our transporter, but theirs has 553 amino acids. I said, are you sure you didn't do something wrong? You know, we went back and forth with why does ours have 554? Why does theirs? We learned that it was a polymorphism. What does that mean? That means that some people have the 554 version, but there are other people who have the 553 version. So everybody doesn't have the same organic cation transport. This had to do with genetic variation. And so that made us begin to think, because at that time, we thought all of this genetic variation and so-called pharmacogenetics was because of drug metabolizing enzymes. That transporters had this variation, that was a little bit surprising to us. So we move it forward. So that we learned also that the variant was a deletion. This one had a deletion at position 420 amino acid position, and it was a deletion of a methionine. That amino acid was gone. So we had a couple questions. How does that genetic variant transporter work? Does it work just like the regular one? And more importantly, and this is what's wonderful about pharmaceutical sciences and what I love the most, is so let's ask the question, how do people who carry this genetic variant respond to the anti-diabetic drug metformin, which is a substrate of alkaline? So this was a question we thought we could now address. So we're in the human genome era, and we're also in the technology era. NIH funded a large network of scientists around the country. And one of the things that NIH encourages has been collaborative research. And these large networks that we're now, Josh and I are now part of another network in Juan, These large networks, science advances very quickly when you have people all over collaborating. So we were all focused on pharmacogenomics. Our group was focused on membrane transporters. We decided we were going to look at all the different membrane transporters and discover the genetic variants in those and figure out how they function and figure if they affected your response to a drug or not. So we had these big dreams, and we were funded for 15 years by NIH. And it was just a great 15 years. So now here is OCT1, the protein that we had cloned a few years before. I guess it would have been about 5 years before. And this is the secondary structure. So all those amino acids that I show you, each one of them is one of these little circles. And they go through the membrane back and forth. It's got 12 transmembrane spanning domains. We resequenced this transporter in 250 DNA samples that we got from African Americans, from Caucasians, from Asians, and some from Native Americans and some Hispanics. So we found that there was the deletion. We found it, and it was present in Caucasians at about 25% allele frequency, and higher in Hispanics, and lower in African Americans. So we found it was ethnic specific, and it was there. But all these red ones shown here, they're not amino acid deletions. They're just a different amino acid. So maybe normally this is a methionine, and then it's a serine in this individual. So we learned there were all these different polymorphisms. So of course that begged a number of questions. If OCT1 has all these polymorphisms, meaning that people in this room carry a different version, not just the deletion, not just the normal form, some other form you may carry, how do they function? Does one function better than the other? Why do I have this one and why not that? So we had a whole lot of questions here. But we also were a membrane transporter group. What about all the other membrane transporters? And at the time there weren't as many as there are now that we knew about, but there were a number of membrane transporters that took up different drugs. And so what about them? So we spent time looking at that, at each of those. But we first characterized the function of each of these variants. So what we did was we created a cell line, we expressed the transporter, and it had this variant alone. And that might have been this one here, serine 14 to phenylalanine. And we measured metformin uptake. So here's metformin uptake in cells that don't have the transporter. Metformin does not get taken up by cells if they do not have this transporter. OCT1 is very important for metformin uptake. Here's metformin uptake in cells that have OCT1, the reference form, without any of these variants, the 554 amino acid version. And then each one of these variants, we made them one at a time, and we expressed them in cells. And you can see that some people carry a form of OCT1 that doesn't function at all, and we saw that. And then some, like the methionine deletion, it functions, but not that well, but it functioned. And then some have a variant that functions quite normally. So during this one, you can imagine how this information was just so mind-blowing to me. Because to me, it said, if that's one gene, look at the variation in one gene, in the function of this one gene, and how it takes up this drug metformin. What about all the other genes? We're beginning to learn about variation between people in how they respond to drugs, how they take up all these different solutes that these drugs take up. So that was a very exciting time. Of course, we asked more questions, and I'll get to those in a minute. This just shows all the different papers. We published many more on different transporters, including Joanne yours. There it is, CNT2, the one she cloned. We looked at the genetic polymorphisms there. We looked at how they function. We began to characterize all this. But in the end, what was interesting to me was, how do people who carry, for example, this variant, maybe they're homozygous, they have 2 copies of this variant, how do they respond to metformin? They're not going to get metformin into their liver because this is the primary transporter which takes metformin and where it presumably has its mechanism of action. So we asked the question of how do individuals who carry this deletion variant and all the other variants respond to metformin? Now this was the beauty of an NIH network. We no longer wanted to do candidate gene studies where we just took people with the genetic variant of OCT1, we gave them metformin, and we took people who didn't have the genetic variant of OCT1, and we gave them metformin. We looked at whether the people who had the genetic variant responded poorly or not. We didn't want to do that. We wanted to do a genome-wide association study. We wanted to take an agnostic approach and measure every single gene that we could get at about 1 million in the genome, and see what associated with response to metformin. And so we did. To do that, we had to collaborate. We had to get at least 10,000 people on metformin. You had to have large numbers because you're testing 1 million SNPs, or at the time maybe it was 500,000 or 700,000 SNPs. So we had to get 10,000 patients. They had to be from one ethnic group because different ethnic groups have different variants in them. So we had to do that. And we published a paper in 2016 in Nature Genetics, and we ended up with 7,000 people. And what was disappointing to me was OCT1 genetic variants did not associate with metformin response, not at all. Now I have all sorts of theories for why that was, but that was not a happy day. But the happy day part was we found something that highly associated with response to metformin. And for me, I was very happy because it was a transporter. It was a glucose transporter, which doesn't surprise me because metformin, that's what it's doing. It's lowering glucose, treating your diabetes. So this transporter is in the liver, and that's what is highly associated with response to metformin. We've replicated this study in another 10,000 people. This is a true result. We found a true result here. We carried out genome-wide association studies for other drugs during this time. And those drugs were allopurinol, an anti-gout medicine, and sulfonylurea diabetic drugs. And in each case, a transporter was associated with inter-individual response in these drugs. ABCG2 for allopurinol, SLCO1B1 for sulfonylureas, and those results have also been replicated. So this was a great era of our lives, and we were very happy during that time. We've now moved on, and I'm now going to get into de-orphaning. So the transporter genome has grown as people have identified more and more transporters in the human genome. We now have, in the transporter superfamily that I studied, called solute carrier superfamily, we have about 400 transporters. They cluster together based upon homology relationships into families. So these are family one. Each one of these is a transporter. So family one looks like seven transporters in family one. Family two has many more. This is in the human genome. Family three has 2 transporters. So each one of the families have transporters there. Most of these transporters sit on the plasma membrane and they move their substrates into cells or out of cells. But some of them sit in mitochondria or lysosomes, so they're intracellular. My lab does not tend to study those. Some of the transporters are nutrient transporters, glucose transporters, amino acid transporters, nucleoside transporters. Some of them really are very promiscuous and take up a lot of drugs. I call them pharmacokinetic or ADME transporters. And they're in there, a whole bunch of heavy metal transporters, or a whole lot of different transporters, vitamin transporters, etc. So we spend the rest of the talk, I will mostly focus on transporters in this family, the SLC22 family, which has a number of drug transporters in it. So we've spent the last 4 or 5 years de-orphaning some of the transporters in this family. When I say that, that means we know the transporters in the family, it clusters together, it has a homology with the other transporters, but no one knew what they did. So we de-orphaned 3 of them, and I'm not going to talk about all 3, I'll talk about one. We de-orphaned SLC 22A15, SLC 22A10, and SLC 22A24. I'm going to talk about SLC 22A24 because I have new data, and so for those of you who have heard me talk before, you will now hear something new because this is now new unpublished data on 22A24. So what you're looking at here now is the 22 family in the human genome. I've color-coded them. So up here in the green are zwitterion transporters. Zwitterions are molecules that have a positive and a negative charge. And so these are zwitterion transporters. One of the transporters we de-orphaned, 22A15, is a zwitterion transporter, that's this one. And we learned that it loves certain zwitterions, particularly the antioxidant ergothioneine. These are OCT1, my favorite transporter, OCT2, OCT3. The ones in black are orphan transporters. We don't know what they do. None of them. We don't know what they do. All of these black ones, this one's black down here too. We don't know what they take up. We've put them into cells. I can tell you, I can talk about that forever. And then the ones in red take up anions or acidic compounds. So I'm going to talk about 22A24, which we de-orphaned a few years ago, but we've learned more about it. And I want to point out that we also de-orphaned 22A10, an anion transporter, which in the human genome is a pseudogene, and we can talk more about that later too. All right, now this transporter to me is fascinating. It's an orphan transporter, but we're losing the transporter. And when I say that, I mean if you look at the transporter, it's just like OCT1, it's multi-membrane spanning. There is a stop codon right here. Normally it's a tyrosine, but it's a stop codon in many people. What does that mean? That means that the whole transporter isn't transcribed or translated, and that stop codon, when it's there, the transporter doesn't work. So if you carry the stop codon, you have a non-functional transporter. Well, look at the stop codon in world populations. It is the major allele. Look at this. The stop codon looks like it's 75, 80% is stop codon. Only 15, 20% of people over here have the tyrosine allele, have a functional transporter. So we are losing this transporter. We don't have much. A whole lot of people don't have the transporter. In fact, because I don't know how many of you have done 23andMe, but we do 23andMe in my laboratory. So we said, let's look up and see who in my laboratory has the stop codon. I was the only one that had a functional allele. Everyone in the lab was homozygous for the stop codon. So we're losing this gene. So that interested me a lot. Why are we losing a gene? What is it doing, and what function is it that we no longer have, or some of us have, and some of us don't have? So we mined genome-wide association metabolomic studies. Now I know Josh and other people I talked to today are really good at doing metabolomic studies, and you learn a lot. That means you measure metabolites, and they measure a lot of different metabolites, 200 to 400 metabolites in the blood of people, and they associate them with genetic variants. So we asked the question, this was Sukwai and I back then, we asked the question, what metabolites associate with that stop codon? What metabolites in people who carry a stop metabolite, don't have a functional transporter, which metabolites are changed? And look at this, androsterone glucuronide. This is the p-value, 10 to the minus 284. For those of you who know statistics, know that nobody's going to argue with you that that is statistically significant. That is a statistical significant association. So sex steroid glucuronide conjugates, 1, 2, look at these p-values, all associated. So that was our hypothesis. This transporter is taking up sex steroid glucuronide conjugates. Now, notice also a couple sulfate conjugates, lower p-values, so we also wondered whether the transporter was taking up sulfate conjugates, and even testosterone itself was associated with a p-value that's quite respectable, just doesn't look so respectable compared to these others. but it was associated with 10 to the minus 25 if we looked at males only. And progesterone was also associated. So we weren't sure about these, but we thought for sure this transporter must take up steroid glucuronide conjugates. It was found in the kidney, by the way. I'll tell you more about that as we go. So what we did was we got the cDNA for the transporter and we expressed it in cells. We threw in androsterone glucuronide and we measured the uptake in cells that had the transporter and in some cells that didn't have the transporter. And as you can see, we got a 270-fold uptake of androsterone glucuronide compared to the empty vector. And for this ethylcolanolone glucuronide, we had a 30-fold uptake, more or less corresponding with these very high, very significant p-values. We did kinetic studies. We learned how the transporter functions. It is functioning as an exchanger with glutaric acid. And we made the conclusion in our first paper that the transporter is a steroid glucuronide transporter. But what about those sulfates and what about the parent compound? Does it transport testosterone? For example, and if you look at sex steroids like androsterone, they're conjugated. They're conjugated with sulfates and glucuronides. And we had found that it definitely took up glucuronide conjugates. Now, why do we conjugate steroids with sulfates and glucuronic acid? Why? Mostly to make this steroid is very hydrophobic, and you add something polar. So we make it more hydrophilic. Once it's hydrophilic, you can eliminate it in your urine and get rid of it. So it is an elimination pathway. So these are more hydrophilic than this. So that was interesting, but how does our transporter function? So as you can see, it likes, this is now uptake. Here's empty vector, which doesn't have the transporter. And here's a whole bunch of different steroid glucuronides, all of them. And it really likes steroid glucuronides, takes them up, what am I at, 30, 90-fold, something like that. Takes up steroid sulfates as well, sulfate conjugates, takes those up. So those are also not as well, but it takes them up. It doesn't like the parent. It doesn't like testosterone, it doesn't like estrogen, and it doesn't like progesterone. Didn't take any of them up. They're about the same as the empty vector. You can see comparing them, maybe a little bit of something there. So it seems to really like the conjugates, but not the parent steroids. So now we thought, okay, we've got a steroid conjugate transporter. What the heck is it doing? We knew it was in the kidney because we looked at RNA-seq data. And we hypothesized that the transporter was on the apical membrane and it reabsorbed back into the body from the urine the steroid conjugates. Now that made no sense. It was one of those paradoxical hypotheses to me. You're making the conjugate to get rid of it, to make it more polar. We're putting it in this way, saying, no, somehow the body wants to reabsorb those steroid conjugates. Didn't make sense to us, but I'll tell you why we made that hypothesis. Looking at data, and we looked at urine data, and here are some urine data, where you're looking at, here's a glucuronide steroid conjugate, here's another glucuronide steroid conjugate. They're highly associated with that stop codon again, but they're associated with the people that have the stop codon have higher levels of these in their urine. So it's as if they can't reabsorb it. They're spilling the steroid conjugates in their urine. So they're getting higher urine levels. So we put it on this membrane and we said it's reabsorbing these steroid conjugates. We then did we made the cell lines in kidney cells that polarize, MDCK cells. We put the transporter there, and you have an apical membrane here and a basolateral membrane there. And then we looked at the uptake of, this time it was taurocolic acid, that's a substrate of the transporter. And you can see we get uptake here compared to empty vector, and that uptake is inhibited by an inhibitor, and that's seen here. But when we add it to the basolateral side, that's the basolateral side, we don't get any uptake. So we do feel it's on the apical membrane, and the transporter functions to reabsorb its substrates, including these glucuronide conjugates, back into the blood. And that's the function that would result in lower urine levels and higher plasma levels. You're reabsorbing it. You're conserving those steroid conjugates. Maybe, why are you doing that? Maybe you conserve it because then you can lyse off the glucuronide and regenerate testosterone. You don't have to synthesize testosterone because you just brought this thing back in, you lysed off the glucuronide, and you've got testosterone, and you could just keep testosterone going that way. So that might be why you do it. For people who have the stop codon, however, they get hot, they spill the steroid conjugate, or in this case taurocolic acid, in their urine, and they get lower plasma levels of the steroid conjugates. So again, the beauty of being in pharmaceutical sciences is you can go all the way to the clinic. And I know many of you, we have many clinical investigators here who take their work all the way to the clinic. So I said, let's just do a renal clearance study, and let's measure the renal clearance of these two glucuronide conjugates. If the transporter is serving in the reabsorption, then filtration, glomerular filtration, and if it's serving in secretion, the renal clearance will be greater than glomerular filtration. So we had to collect a lot of different samples, urine and plasma, and we managed to get, 70 or 80 samples here. Most people have the stop codon. These are people that don't have any functional allele. We only found 7 people. I volunteered to participate in the study, but I didn't, by the way. They said no, they didn't want me anyway. But there were 7 people that had functional alleles. They may have only had one, but they were functional. And we found their renal clearance was less than these people, but there was a lot of variation. And we found it for ethylcolinolone and androsterone glucuronide. When we compared the renal clearance to GFR, sure enough, the renal clearance was less than filtration clearance, indicating, and these are the people that have the functional allele, that it is reabsorbing. The renal clearance is just, it's taking it out of the urine, and you have a less than GFR clearance. So that was good. That went along with our hypothesis. But here's what you have to do when you're a true scientist. You look at that data, and you stare at the data, and you say to yourself, yeah, that's true. It is. There's only 7 of these guys. And yes, it is. The renal clearance over here is less than filtration, so it is being reabsorbed. But what about these guys? They don't have any functional transporter. They have 2, they are homozygous for the stop codon, but they're doing the same thing. So I sat there, and look at this, it's even more dramatic for androsterone glucuronide. So I sat there and I told my students, they said, oh, look, we got all these great data, and they showed me that. And I said, yeah, but what about these guys? They don't have a transporter. So it's hard for me to conclude that our transporter is a major transporter when it's not. So we made 2 hypotheses. And science is always driven by the unexpected, I feel, and the most interesting things are the things you're not expecting. So I said, cheer up, we're going to figure it out. So one thing we did was, a lot of drugs, as you know, people who have studied pharmaceutical sciences know that a lot of drug molecules are passively reabsorbed in the kidney. And so this is the glucuronide conjugates we're talking about. It was hard for me to imagine they were passively reabsorbed. But I looked at their oil-water partition coefficient. So here's androsterone. That oil-water means you put a little oil on the top, you have water on the bottom. Where does the drug go? So as you can see, androsterone alone, 5,000 to 1. It's definitely in the oil. Likes the oil. It's what you call highly hydrophobic. But I was surprised to see that the glucuronide conjugate was 31 to 1. So there might be some passive reabsorption. It may cross the biological membrane, and there could be passive reabsorption. The sulfate looks like you need a transporter for sulfates. It's more polar at 2 to 1. So I got to prove it. I only speculate that we can be reabsorbing these glucuronides. We don't really need a transporter. We saw all those people, they don't have a transporter, and they're reabsorbing it. That said, we have the transporter, and could there still be another transporter? So we went to, this just shows, here's a nephron, and this just shows that, this could just be passively reabsorbed, the glucuronide conjugates. All right, but we looked at the family anyway. And there's another transporter on the apical membrane, OAT4. So here's the transporter we've been studying, 22A4, which has 22A24, that's a typo. Here's a transporter that we've been studying that most people don't have anyway. And here's OAT4. And OAT4 is right here. It's called 22A11. And it's very close in homology to 22A24. And we know it sits on the renal proximal tubule. And we know it reabsorbs substrates. So we said, let's see if it reabsorbs or if it transports glucuronide conjugates. So we tried, in this case, estrogen 17 glucuronide. Here's SLC 22A24, takes it up very nicely. But here's OAT4, 22A11. It doesn't take up this glucuronide conjugate at all. So we tried, I said, okay, let's try every glucuronide conjugate we have in the lab. So we tried every glucuronide conjugate. 22A11 doesn't take up any glucuronide conjugates. So I can only think that those glucuronide conjugates in the people who have a non-functional SLC 22A24, which are most people, might be just being passively reabsorbed. We then looked at sulfate conjugates, by the way, and it looks like both, let me see if I've annotated it now, both 22A24 and OAT4 take up sulfate conjugates. Here's the empty vector, here's 22, here's OAT4, and here's 22A24. 22A24 seems to do a better job than OAT4, but OAT4 takes up sulfate conjugates. So they do take up sulfate conjugates. Now, when you look at the location in the kidney, and this is really going deep, is you see 22A24 is found in the proximal tubule, but it's found in segment 3. That means it's down here in the proximal tubule. Whereas OAT4 is all over the proximal tubule, segment 1, 2, and 3. So it's everywhere. When I see a transporter just here in the proximal tubule, I think it's a cleanup transporter. You already reabsorbed everything, sulfates. You reabsorbed them with OAT4. You're reabsorbing, and then finally just a little bit is left in the tubule. You don't need anything but a cleanup transporter, and a cleanup transporter will likely have a higher affinity. So we looked at that, and what we found was if you look at the Km value for these steroid sulfates, here's OAT4, here's DHEAS, 250 micromolar Km, whereas for SLC 22A24, it's 7 micromolar. So we think what's going on is OAT4 is, for glucuronides, the only transporter that's working is SLC 22A24, and you're passively reabsorbing them, probably at a slower rate, but you are. For sulfates, OAT4 is working all along, sulfate all along the nephron, and down here you don't need much. You just need a high affinity transporter to clean up the rest, and 22A24 and OAT4 are down in segment 3. So that's what we think is going on biologically. Now, I get into grade 8s a lot. I get into monkeys and I get into evolution. Why? We're looking at a transporter that we're losing. So I started thinking to myself, well, why did we lose it? When did we lose it? And who lost it? What about Neanderthals? Do they have this dot codon? What about them? So we started with with OAT4. So my hypothesis on OAT4 was that nobody's losing it. It's a workhorse. It's found all along the tubule. You need it for sulfate conjugates. They are not nonpolar. And so sure enough, bonobo, this is GFP tag, shows it's on the plasma membrane. It functions very nicely. This is estrogen sulfate. Chimp, yes, it takes it up. Orangutan, gorilla, it's taken up. And it's highly conserved, and we don't see any real polymorphisms there. We certainly don't see stop codons in OAT4. The other thing is, when we look for OAT4 in all mammalian species, which we look when we search for the genome, There is a direct O4 ortholog for every single mammalian species that we can find. For SLC 22A24, well, the bonobo has two versions. One that has 522 amino acids, one that has 534 amino acids. They're missing big segments of amino acids, and it's not on the plasma membrane, and neither version functions. The chimp has two versions. One is missing an amino acid, and that one doesn't function, and the other version functions okay. And then orangutan and gorilla, we don't have a lot of data in the database to know if there are stop codons, but they have functional alleles. And of course, people that have the tyrosine allele like I do, we have a functional transporter, but most people, they're down here like the bonobos also. So this is a transporter that we are losing in evolution. And so we went and looked. We have a wonderful collaborator, Tony Capra at UCSF. He likes to look at ancient people. So we found a lot of ancient bodies we've read about over the years. They've gotten their DNA, and they've sequenced those guys, and they're up on databases that Tony knows about. I don't know how to get to them. So all the ancient people. They also have sequenced Neanderthal and Denisovan. These are hominids that are related to us. We're Homo sapiens, but they're not us. So we found, or Tony found, that the stop codon existed in Denisovan, it existed in the Neanderthal, and of course it exists in us. I put the stippled line because Denisovan, Neanderthal, and us also have the tyrosine residue. So some people have the tyrosine, and some of these Neanderthals did as well. The chimp, as I put it this, it's sort of there in some chimps it's not. We're seeing it in these other great apes. Old and New World monkeys, we don't see the transporter, they've lost it. And most mammalian species don't have 22A24. A horse does, certain ones do, but not too many. So it is a gene that is being lost. So summary on this, genes with common loss of function polymorphisms in human and loss of function in primates and many other species. This is 22A24. O4 gene with very few polymorphism. All orthologs are present in all mammalian species, and we don't see any non-functional alleles there. There might be a rare one, but nothing common. The 22A24 is broadly selected for sulfate and glucuronide conjugates. O4 has a narrow selectivity for sulfate conjugates. It doesn't take up glucuronide conjugates. This 22A24 has a higher affinity. It's a cleanup transporter, and the renal proximal tubule, and is a lower affinity transporter. And AlphaFold does what it should do, and that is predicts that this transporter takes up more compounds, and this one has a narrower binding capacity. All right, last little segment, and about 4 slides to go, and I think I'm doing okay. I'm just going to show you the work we're doing on the human blood-brain barrier. So we've been interested over the years, and people here like John are interested also in the blood-brain barrier, the blood-CSF barrier. We're funded in a grant, which is part of a network. I think I showed on the next slide. These are my collaborators at UCSF who are working in this study. The network is called the Transporter Elucidation Network. People here are studying the placenta, the mammary gland, that's at Columbia, I believe. They're studying mammary gland. Johns Hopkins is studying mammary gland. Intestine is here, and also Rutgers, are they doing intestine? Hopkins is also doing that. Okay, and then blood-brain barrier we're doing the blood-brain barrier. So we're looking at the transporters in the blood-brain barrier and asking questions about what's there. How do things get across? So let me tell you a little bit about the blood-brain barrier. So the brain is a protected organ, meaning that normally capillaries are leaky. Things can go between one cell and another, there's a space, and things can kind of leak in and out of capillaries. But in the brain, the capillaries form tight junctions, and they're not leaky at all. So things can't just sort of float into your brain very easily. And that's because The capillaries form these tight junctions. This is a cross section. This blue are the capillary endothelial cells with their tight junctions. And around the capillaries is something called a pericyte. And then there are neurons, shown here. And then there are astrocytes. I think this is a podocyte. And there's a basement membrane and a pericyte here. This is the complicated sort of structure that forms those capillaries that prevents a lot of leaking into the brain. But the brain needs nutrients. The brain needs glucose. The brain needs amino acids. So you need to selectively allow those to go in. So there are transporters there. And so we took it, we set it on ourselves to what transporters are there, and do any of them interact with drugs, etc. These are some of the questions we're addressing. So we got brain samples from the NIH Neurobiobank, and we got them from 0 to 24 a month old, from children greater than 2 and all the way to 60-year-olds. We call those adults. And then for greater than 60, we just labeled that elderly. And we prepared the brain microvessels, which means we homogenized it, we extracted, we purified it, and we spent about a year trying to optimize these methods so we didn't have contamination with neurons or other things. We wanted the capillaries themselves. And we got a pretty good prep in the end. And we just published the study, and I will show you some of our data. So first, I'm going to need water. Let me see if I have enough. So first, we use global proteomic method, so I'm not showing you the whole story here, but I'm showing you the transfers. So highly look at the